Welcome to Concordance Answers. Since this is our very first episode, let me explain. I'm going to be answering one science question per week from a user-submitted question. You can submit your own science question through the polling feature on my channel. This week's question comes to us from Spylab of Athens, Greece. They ask, how did 24 primate chromosomes in the ancestral line of humans become 23 chromosomes in the human line? Some of you may be aware of this, but 2 of the primate chromosomes in the ancestral line of humans fuse to become 23, so that all of your diploid cells contain 46 total chromosomes, whereas a chimpanzee would have 48 total chromosomes. There are a couple of ways I could explain this. The short answer is probably a Robertsonian translocation. We don't know exactly what happened because it was a historical event that we can't recreate. However, I've decided to go to the lab and use our most scientific tool to explain how a Robertsonian translocation could result in the loss of a chromosome. I have here 2 pieces of Play-Doh that are going to represent chromosomes. Now, chromosomes are our genetic material. They contain all the genes, and they have certain structures to them. At the ends, they'll have telomeres, and in the middle, they'll have centromeres. The telomeres help to keep the ends from unraveling in a very simplified form. And the middle, the centromere, provides a site for attachment. This is a very specialized structure that the cellular machinery attaches to in order to drag these chromosomes around. Now, during meiosis, they line up. 2 sister chromatids will line up. And recombination will occur. And I'm going to simulate recombination occurring here. Right? We have a crossing over. We'll do it as sort of a pinching off. In fact, the 2 strands sort of break and then are reformed. So that what was once here is now over here. And you can see that the cell does a little patting and massaging of the Play-Doh chromosome, and it's back together. So that's normal crossover recombination. Occasionally, the wrong chromosome will pair up, and you'll end up with recombination. This is called a translocation, where part of the first chromosome, the genes that were on this chromosome, are now part of this chromosome. This almost always leads to some form of genetic disease. Sometimes we have a balanced translocation. That gene's gone. We have a balanced translocation where the genetic content doesn't change, but whatever was here at the junction has been disrupted. So if there was a gene that was in this region, it's probably damaged and non-functional, and that can lead to genetic disease for the children that are born from this genetic complement. Sometimes we have an imbalanced translocation, and that results in a net change in the genetic complement, so that we've lost genes or we've even gained genes, where we can end up where we have multiple copies of the same genes being present. So now we're going to talk about a special case of translocation. This is translocation between 2 acrocentric chromosomes. Acro means extreme, and then the centra refers to where the centromere is positioned. So in this case, our centromeres are positioned very near the end of one end of the chromosome. So the long and short arms are very asymmetric. We have a center point that's very near one end. Now when the translocation occurs, the short end of each of these molecules is lost, and recombination occurs here in the middle, and you have essentially very little loss of genetic information, but the fusion of 2 chromosomes. So now we've gone from 48 to 46, but the total number of genes hasn't changed by much. We've probably lost these 2 short ends, but they may not have contained much genetic information to begin with. So how common is this? Well, for every 1,000 live births today, about 1 child is born with a Robertsonian translocation. The rates are higher in other species. The phenotype or appearance of these children can be anything from normal to severely disabled. It is quite possible that 2 primates with the same complementary Robertsonian translocation met and mated, and we ended up with us.